Okay, so I guess I'll start over. So those who, or I guess they just couldn't see my slides. So I will continue. So given the prevalence of cardiovascular diseases worldwide, many have attempted to determine the genetic underpinnings of disease progression and outcomes using GWAS. However, the patient population, the heterogeneity, sorry, I'm just trying to get is there a laser pointer or should I just use my pointer?
No, yeah.
Okay, I'll just use the mouse then. However, the heterogeneity in the patient population, environmental factors, and complex, move it on.
Put down. Yeah, there you go.
There, that's better. And complex nature of diseases like myocardial infarction and heart failure have made it really challenging to link them to the genome. Similar studies have been done in the mouse, where we can better control for environmental factors and genetic diversity. However, the challenges with performing experimental MIs still limit the ability to identify relevant loci. And this study was only able to identify one locus that was associated with the prevalence of cardiac rupture, which is an outcome that is very rare in the human patient population. So one approach we can use to bypass these challenges is to map using surrogate phenotypes or traits that are predictive of disease outcomes. And this example was done by Dr. Patterson. She was able to map the frequency of mononuclear cardiomyocytes across the hybrid mouse diversity panel, was able to identify a gene that was related to this phenotype, as well as confirm that the surrogate is indeed impactful in terms of outcomes after MI. Similarly, I'm also interested in how cardiomyocyte ploidy is going to be influencing cardiac disease traits. And one thing that I just want to point out before moving forward is that the majority of the adult mammalian heart is composed of these tetraploid or 1x4 or 2x2N cardiomyocytes. And the tetraploidy arises when a cardiomyocyte enters an endomitosis, which is an alternative cell cycle, and it fails to complete mitosis, so developing two diploid cells. Now, the phenotype that I'm going to focus on today is one of the two that we are planning on mapping, and what we are calling the hyperpolyploid cardiomyocyte population. These hyperpolyploid cardiomyocytes have 8N or greater DNA content. There are a lot of theories surrounding why a cardiomyocyte would become hyperpolyploid. One of them being that having more DNA content allows them to synthesize new proteins faster and therefore mount a compensatory response under injury conditions faster. On the other hand, higher frequencies of this higher polyploid cardiomyocyte class are found in patients with end-stage heart failure and dilated cardiomyopathy. So there's still much that's unknown about this, why cardiomyocytes would become hyperpolyploid. And I'm really looking to investigate how the frequency of this class influences baseline cardiac function, as well as outcomes after myocardial infarction, or MI, which I will use going forward. And then, so the mapping portion of this project will be using the Hybrid Rat Diversity Panel, or HRDP, as many of you know. So briefly, this panel is composed of 2 recombinant inbred panels, as well as 33 classic inbred strains that have been compiled to maximize the ability to map complex traits. So far, I have been able to quantify the frequency of hyperpolyploid cardiomyocytes across 72 strains, ranging from, it was a lot of work, like 2 years worth. But ranging from 0.8% to 20.7%. Notably, this phenotype has a broad sense heritability of about 89%, suggesting that this phenotypic variation is largely due to the genetic variation in these strains. So the first thing that I really needed to do was to confirm that this is a appropriate surrogate phenotype for the cardiac disease traits we're interested in looking at. And to do this, and actually, I just want to make a note real quick. The bars that are colored are the strains that are moving forward in the studies of the cardiac physiology. So spanning across the entire range. So to do this, we perform a permanent ligation of the left anterior descending artery. And then we assess cardiac function by echocardiography. And we do this before we perform the injury, 3 days after, and then at 28 days, where we end the experiment, euthanize the animals, and collect tissue for further analysis. So I was excited to find that there were indeed correlations here with left ventricular ejection fraction, which is a clinically relevant measurement that is basically looking at the amount of blood that is being pumped out of the heart each time it beats. In addition to this correlation, there were also correlations with the left ventricular area, so the amount of space that's within the chamber. And also with left ventricular mass, which is just a measure of the left ventricle weight. So altogether, these baseline correlations could indicate that those strains that have higher frequencies of hyperpolyploidy have dilation of their left ventricles with reduced contractility. And these are things that are hallmarks of dilated cardiomyopathy and precursors to heart failure. So as far as our MI studies go, we also were able to see a correlation with hyperpolyploidy and the change in the LV area, which is just basically a measure of dilation. So how much have from their day 1 or baseline or day 0 and to day 28, how much has that ventricle expanded? And as you can see, the strains with lower hyperpolyploidy had the tendency to expand larger at the 28-day time point. And this could really indicate that there may be some protection to adverse cardiac remodeling following myocardial infarction when you start with a higher frequency of hyperpolyploid cardiomyocytes. So now, knowing that our surrogate phenotype is predictive of some cardiac disease traits, our next step was to perform some GWAS, so mapping with our collaborators from Colorado, and Dr. Lewis. And so at the time, our previous mapping, there were 63 of these strains that had genotype data available for mapping. So our preliminary results, although we did not reach any statistically significant peaks yet, there are 6 suggestive peaks. And this could just mean that at a lower level, there are multiple genes contributing to the regulation of this phenotype. And so I just want to point out what I'm going to be looking at a little closer going forward is this chromosome 14 locus, which has the lowest p-value and is what we will be pursuing for candidate genes initially. I do want to mention before explaining this that we are taking a relatively limited approach here and do in the future plan to interrogate these loci with more rigor. But as of right now, we wanted to just start by looking at within the protein coding genes in these or in this locus, are there damaging or potentially damaging variants that are found in the protein coding genes. So within this locus, there are 151 protein coding genes. Of these, there are 22 that have variants that are predicted to be damaging. And then when we look at cardiomyocytes specifically, 7 of these are found to be expressed in the mouse and human cardiomyocytes. And one in particular that I want to highlight here is SHRM3. Its role in the body is to help regulate cell shape. And when we look at using tabula muris at the expression in the heart, it is fairly restricted to cardiomyocytes. So that definitely piqued our interest. Additionally, so SHRM3 has 7 protein coding variants that are potentially damaging. And the ones that I have in these red boxes have actually previously been functionally characterized in chronic kidney disease by people at MCW previously. So these red variants, 5 of these are actually going to be found in HRDP strains. And in this study, they were able to basically bring it down to just this G1073S variant in the actin binding domain as a variant that was damaging in the kidney. And this was because this variant was able to reduce the actin binding affinity. So when we look at the strains of the HRDP that have this G1073S variant, we found that the strains that have higher hyperpolyploidy also more often harbor this S at the 1073 position. So going forward, we would like to test SHRM3 in 2 different in vivo models. The first, because it's shown to be functionally important in the kidney and to affect kidney function, we wanted to ask if there is a cardiomyocyte intrinsic role for SHRM3. And so we're doing this with our collaborators in Dr. Caitlin O'Meara's lab. They are testing some conditional knockout mice, cardiomyocyte specific conditional knockout mice for SHRM3. And we're asking, this is not the most current version, that's okay. Ignore question 3. So we're asking if the loss of SHRM3 in cardiomyocytes specifically will increase cardiomyocyte hyperpolyploidy, and also what will it affect baseline cardiac function and myocardial infarction outcomes. Our second model that we are making with the help of the Rat Research Model Service Center at MCW is an SHR rats that will have this G1073S variant corrected back to the reference genome allele. And we'll be asking basically the same questions, but now instead focusing on whether this reduced binding affinity that this variant causes alone is enough to influence these phenotypes. So with that, I just want to thank all of the people who have supported this project, as well as the funds that have made it possible. And with that, I would love to take any questions if we have time.
If you have time.
Perfect.